Hello, I have no conflicts of interest, and this is the grant that has funded my research since 2000. I couldn't believe it. I got a PhD and had an idea, sent it into the NIH, and they gave me $1 million. It was like, the poor black girl from Los Angeles got $1 million from the NIH, and this was just incredible to me. And we've been working on this grant ever since. So I'll tell you a little bit about the things that we have learned along the way. And then, yes, you can take photographs, it's fine. Just don't sell them, I guess. Not that you would. All right, so this is my first introduction to the field of sleep in 1988. Michael Chase, down in the center, organized this basic sleep training camp that was held at Lake Arrowhead for about a week. It was really for trainees to increase the field, because pretty much in this picture was the total field of sleep at that time, at least basic sleep research. It was small, and so just as was introduced earlier, how the number of publications have grown by a factor of 24, so has the number of people doing basic sleep research over the years. It's not that we're all just that much more productive. Instead, there's just many more of us. And I'm so gratified to see all of you here to advance sleep. If you should think for a moment that your work is not important, please think again, because sleep is the reason we have life, and the reason we can make choices and pursue happiness. So thank you to all of you in this audience for really what is an essential life-preserving line of work. I'm so glad you're here, and I'm so glad to be here myself. Spread the word so that there can be even more of us, because even though we know a lot of the functions of sleep, we actually don't know all of them, and we don't know every animal that sleeps either. So, all right, let's keep going. There we go. Okay, yeah, back. There, I took my postdoc at the University of Arizona working with Bruce McNaughton in the middle there, and Jim Knierim, who's to your left. And these guys, we sent 4 rats up into space in the space shuttle. So I put this in here because we're in Houston, and I spent a lot of time during those years in Houston working out the research kinks so that the project can go smoothly. And these guys were really great to work with. It was Neurolab's Space Lab mission in 1998. And this is the crew. I almost made it onto the crew. Yeah, so you can see on the right, there's a picture of one of the astronauts in a sleep cubicle, because there were lots of human sleep research going on at that time too. And it's been a great tradition in the sleep field. We don't just stay on Earth, we see what zero gravity and the heavy workload that the astronauts participate on board does to our circadian rhythms and our ability to sleep. And this is this project we've set up. It's called the Escher Staircase, and what it is, it's a maze that a rat couldn't run on Earth because they have to run up walls and turn right. And with only 3 90-degree yaw turns, normally it takes 4 to get back to where you're facing, but in space on this Escher Staircase, only 3 yaw turns, they could get back to where they started. And we hypothesized that they wouldn't be able to map this environment correctly. I had a secret hypothesis in the back that, in fact, they would learn to navigate this correctly by rewiring their nervous system between the first and the second time we ran them on this maze. Indeed, the very first time we ran them on the maze, their place cells, the cells that encoded where they were on the maze, were all messed up on all 4 rats. But by day 9, when they ran it again, the second time they ran it, they were perfect. The place cells were perfect. So we have recorded, so what we did after we ran them on the Escher Staircase, which actually was kind of hard to do because the rats loved being in space, and they let go of the Escher Staircase a lot and floated around and did flips, and it was really nice to see, but it was also, you could see the hand of the astronaut putting them back on the maze. They were stimulated directly in their pleasure system in order to run this maze,
and still they preferred to let go and explore the space. So that shows you how pleasurable it is to be weightless and floating around. All right, so yeah, by day 9, it was perfectly normal, their place cells. And what the astronauts did after the rats ran this maze is put them in a little sleeping pouch, a little sleeping bag, zipped them into a sleeping bag so that they would relax. And as they did this, we kept recording their sleep, the neural activity during sleep. Unfortunately, I have never been able to get that data to analyze it, but I hypothesize and think that what happens between day 4 and day 9 in the first 4 hours of sleep is that they actually said, I don't know how to map this environment, let's see what we can do. And they, through REM sleep and transition to REM sleep, which is N2, really rewired their hippocampus. And I'm going to show you through other experiments why that might be true. Okay, so they did sleep after the run. Did it allow the new map to form? We don't really know, but hopefully that was what happened. So we're going to talk today about features of sleep that serve learning and memory, gains of insight, and our emotional health. So we know a lot about sleep now. Back when I was 1988, we had lots of hypotheses, but not that much evidence or very many people, and it was a young field. But now we know that sleep can serve to clean the brain. There was a recent article that may counteract that, but I don't think so. They use dye. We could talk about that later. But it also restores the immune system, we now know. There's a big bolus of growth hormone release that we actually knew a long time ago, but the function of that for forming proteins has been studied. A really nice paper in '89 and another one very recently showing protein synthesis goes on quite rapidly in sleep. And then energy restoration, temperature regulation is something we knew from a long time ago. Actually, that was the first thing I'd ever heard about sleep, is that homeotherms like us stop thermoregulating in REM sleep, and that's really weird and really dangerous. Why would we do that? I'm going to show you how important sleep is across species, especially REM sleep. And also chromosome repair. Now that, to me, seems like maybe that is the essential function of sleep, chromosome repair, because in fact, our chromosomes get unraveled and broken. Our DNA gets broken every single day, and it's amazing. But during sleep, there's great studies showing we repair that. What would we do? How long would we live without repairing our chromosomes every day? I think that's the Nobel Prize winning. It's not something I work on, unfortunately, but it's going to be the Nobel Prize winning one. So we prune specific connections. I'm going to talk about that and how we've revealed that in specific circuits. And this really shapes our nervous system, both while we're developing and throughout our life, as long as we're learning. So what is sleep? Well, we all know what it is, right? It's a behavioral state. That's how it's defined. That's homeostatically regulated. If we don't get enough of it, we try and get more later with a characteristic posture, which is not this. I didn't snap this picture, but I could have. One of the most sleep-deprived nations, Japan, you will find people sleeping on the train all the time. And this guy really needed it because he wedged himself in. Can't imagine how his left hand or right hand is going to feel when he wakes up. But yeah, so that's not the characteristic posture. But we have reduced responsiveness to outside stimulus, and we have an increased arousal threshold, and thankfully it's rapidly reversible. And it's also characterized by typical electrophysiological traits, which I know all of you know, but I'm going to show you a few of them, and neurochemical milieus, which I don't know if all of you know about that, so we're going to stress it. So this is a compilation of photos of different animals sleeping to show that all animals sleep, from whales to worms and even humans. Fruit flies, I'm going to show, there's a fruit fly buzzing around, as we just heard from, let's see, David Risen. Fruit flies sleep as well. In fact, I'm kind of envious of anyone who's been able to study sleep in invertebrates, because There was a recent paper in C. elegans that basically replicated my last 30 years of research in one paper. And so I was like, why didn't I take that postdoc at the University of Pennsylvania where I could have studied that? Anyway, so from whales to worms, we all sleep. And we used to think, really, when I was going through grad school, that only birds and mammals had two types of sleep, that all other animals just had non-REM sleep, slow wave sleep, and that was it. But now we know from more recent evidence that lizards have rapid eye movement sleep. This is a dragon lizard. And then fruit flies, even they have two states of sleep, one that's more active where their limbs are twitching, and one that's quiet. And jellyfish, okay, jellyfish, David was talking about, these are the guys at Caltech. They showed that jellyfish do sleep and it's homeostatically regulated and there's a typical posture. And they even have in one of their figures, I think it might be figure 2, two states of sleep, which I didn't really talk about. Here's a water bear, which is a microscopic organism. We don't know if they sleep yet. No one studied it, but it looks like it's sleeping, so I left its picture in here. So cute. All right, so this is a video taken from the ship.
Oh my God!
Oh, that's so loud, sorry. She was really scared because you'll see in a moment when she pans to the right, she came across a pod of whales, or the sailing ship came across a pod of whales that were there in this position, right?
Okay, I'm really scared.
You've never seen a whale. I've never seen a whale in this position, but there's a whole bunch of them straight up and down looking like aliens.
Okay.
And
Dad.
this is the state of sleep that no one's ever recorded the EEG of a whale. It's hard to get down past all that fat, but this is another state of sleep of whales that hasn't really been studied very much, but I will show you in the next a beautiful study published in 2008 that shows that whales will, all over the world, will do this, where they will swim down. In black is the depth of the whale from the surface, and blue is the pitch of the whale, so it's straight up and down, you can see in the first 20 minutes or so. And the speed in red, which is just 0, so it's not moving at all. And then you can see that for about 15 minutes, the whale's in that straight up pitch, not too deep from the surface of the water. And then it goes horizontal for about 10 minutes, probably breathing and awake. And then he dives down head first, way down to almost 30 meters deep, and stays there for about 5-10 minutes, and then just drifts actually to a head up position. So it's completely neutral, and kind of drifts along for another 20 minutes, not breathing, and finally wakes up and swims to the surface again. And whales all over the world do this. You can see in different seas, the amount of that completely neutral sleep is in yellow. So that's in an inactive dive, it's called, in yellow, across the different areas. And it varies from about 8 or 9% to about 2%. In the Mediterranean, whales swimming into the Mediterranean only have this neutral sleep, which is like REM sleep because they are completely atonic. It's so important that they will stop breathing for 20 minutes in order to get this state, and all over the world. I don't know why, no one knows why whales in the Mediterranean have much less of this neutral sleep than other whales in other places, or the same whales in other places. But it would be really interesting to see, if you want to study it, whether it's just ship traffic or something else that doesn't allow them to get as much of this state as in other places. All right, there's a lot to learn. Okay, so here's Sleep in C. elegans. This was a really great paper published just last year where they teach worms, to your right is worms that have been taught by a series of exposures to what is one of their favorite food smells. And just they're given that food smell, but never given any food to eat while that food smell is there. And after a series of those and some sleep in between, 2 hours of sleep opportunity, they will learn to, when exposed to that same smell, just not go to it anymore. Normally they would run or squirm to that food. But after a period of training, they will learn that smell is no longer associated with foods. They won't even bother going to it. So that's what it is, worms around the outside, not going to that smell in the middle. And they found that it's really sleep that's important for them to learn to forget that great association between that smell and food, because they did sleep deprivation. They eliminated a type of neuron that's really important for sleep, and they found that the animals didn't learn it if they did that. They lost the memory that they had built up in the waking period before, that sleep was no longer, that food is no longer associated, that smell is no longer associated with that food. Really cool paper. That's the one that kind of replicated my 30 years of research and did it even better, because they were actually able to show the reduction in the number of synapses that occurs only if the worms sleep. So they have synapses between the delicious smell and the go get it system, and that the synapses between those systems reduce in the sleep between the learning and the performance. And that's what my research is all about too. So Jeff Donnelly and I did a review of this, a commentary on it, just showing that the neuron, AWC, that's the delicious smell kind of neuron, connecting to the AIY, which is go get it, and the ALA neuron on the top is a sleep active neuron. And if they lesion it, they don't get this. But anyway, once they learn in that triangle number 2, that smell is no longer associated with good food. During wakefulness, the synapses are still there in the first learning period, but then once they go to sleep, and this is their sleep posture, a hooked posture, then you can see that the number of synapses degrades only if they're allowed to sleep. And so the next day next waking period, when they test them, they don't have many good connections between those two areas of the brain. Just the coolest study. All right, so let's see. So that's how we are not confused. This is how sleep rewires our nervous system, so we're not confused about the next day about the things that we've learned the day before. We are integrating it into our nervous system, pruning back those things that need to be pruned, strengthening those things that need to be strengthened. So across phylogeny, from whales to worms, all animals sleep, and we know if you don't sleep, you will eventually die. We know this is because of fatal familial insomnia disorder. We know this from research in animals, that we die, and what we die from seems to be varied. I'm going to call it multiple systems failure, because sleep is so important for all of our systems. And I talked about the immune system, that's one of them, metabolism, chromosome repair, all of those really essential functions. So I'm going to go back to some things that you all know, which is just to make you feel comfortable again, which is that Fitbit doesn't measure EEG. That's all something we know, not yet. Hopefully the technology will change, but so I just overlaid some pictures here. So stages of sleep, we go from wakefulness in yellow to N2 to deep slow wave sleep, where we have these big slow waves going through our brain. Then we go to the transition to REM or N2 state with sleep spindles, and the density of those is really well correlated with our IQ, but they last about 1.5 seconds, and they come a few times every minute. And then we go into REM sleep, which is characterized by a beautiful theta rhythm, which is 5 to 10 hertz across species, and is more big and more beautiful in REM sleep than it is even during wakefulness. The other time we have theta in our hippocampus, our learning and memory structure, is when we are learning and remembering. So hopefully all of you have some theta in your brain right now. And then when we're in REM sleep, we have it even more steady, bigger, and more beautiful. So this is the amount of sleep we get throughout our lifespan. This is my grandson, he was only a month old at the time, at that active sleep, which is what we call it when we're infants. And you can see on the bottom of the graph, there's from weeks to months to years. And the amount of REM sleep goes down and down and down as we get older and older, as does the amount of slow wave sleep. The N2 state, thankfully, stays fairly steady across our lifespan, although it goes down a little bit when we're older. Yeah, many parents, I think, mistake active sleep for wakefulness because, well, the eyes are sometimes not all the way closed and they're moving and they're doing a lot of motion. But never wake a sleeping baby, and I'll show you why. All right, so this is a beautiful study by Elizabeth Clareman and colleagues that replicates 2 other studies where they just put people in the most boring room possible. There's a bed, nothing else. They can't do anything else, and she does it for 14 hours a day. Is it 14 or 16 hours a day? Okay, 14 to 16. The gray is 14 hours a day and the purple is 16 hours a day. And she gives them nap opportunity for 2 hours in the middle of the afternoon. And you can see that on average, people level out to about 8.5 hours a day, or even more, 8.9 in one of the studies. So that's almost 9 hours a day. Who in this room regularly gets 9 hours or 8.5 hours of sleep per night? Oh, I see like 1%, 2% of us. Congratulations, good for you. We should all be looking at them for everything because, an earlier study by Tim Rears showed this to be true also. He did a study for 1 week and another one for 2 weeks, and he showed they leveled out to about 8 hours and 15 minutes a day, but he only confined them to this bedroom for just 12 hours a day. And he asked them at the end how they felt, and they felt great. How great do you feel if you're able to get this much sleep? And think about all the multiple organs that benefit from sleep. We should really be trying to encourage people to get this much sleep every day. Not 7.5 hours, 8.5 hours. That would be amazing. All right. So this is a study, the most cited study, the most picked up by the media study that was done by postdoc Cow et al., Van Savage at UCLA, asked me to come help comment on this study and be part of it. This was picked up so much by the media because the title that the media's put on this was, "Scientists discover why we sleep." That was really embarrassing, and I hope you guys don't hold that against me. I did not say that, but this is one of the many reasons why we probably sleep, and that is in the first few years of life, on the top is years, our brain is undergoing enormous reorganization. We have many, many synapses, and experience prunes some of those synapses and strengthens others. And you can see the total amount of REM sleep compared to the total amount of sleep, and that's on the y-axis. There's a natural log of this. Elizabeth Clareman will know what this means better than I do. But reduces as our brain starts to organize. There's age 3, at which point that beautiful correlation goes away, and it's more the amount of slow wave sleep that is associated with changes over the lifespan. So here is my nephew learning to walk, and it's just one of the many examples of the things that we are learning in those first few years of life, which is probably why we have so much REM sleep. Okay, so let's turn to the neurotransmitter milieu, and this is just to show you how different one state is from the other in the brain in terms of the neurotransmitters that are swimming around. So in the first column to the left is wakefulness, where you have lots of acetylcholine. Hopefully, again, you guys have acetylcholine swimming around in your brain, a lot of it because it seems to be like the searchlight of attention. We have lots of norepinephrine from our locus coeruleus, lots of serotonin. Our serotonergic neurons are very active. When we go into our deep slow wave sleep, the one thing that has to change is the amount of acetylcholine. That goes to 0. You can see during slow wave sleep, acetylcholine, the basal forebrain cholinergic cells that are responsible for that searchlight of attention, shut off. They're actively inhibited, and that releases the thalamus to produce these big slow waves. And then we go into that N2 transition to REM state with beautiful sleep spindles, and you can see all these neurotransmitters are off. It's an incredible state. It's really the opposite of wakefulness if you think about it. All of these neurotransmitters are off, and they need to be off in order to produce these sleep spindles, and I'm going to talk about the real importance of these sleep spindles to the transfer of memories to the cortex in a moment. but you can see that's super important. And then we go into REM sleep with that big, beautiful theta at the top. And you can see there's even more acetylcholine swimming around in the brain than during wakefulness when we're learning, and no norepinephrine and serotonin. That's these brainstem nuclei that provide them to the forebrain are actively inhibited and shut off during this state. All of these neurotransmitters are responsible for different things. Acetylcholine, searchlight of attention, allows our neurons to be plastic. It sets up that theta wave, which is really important to learning. And then norepinephrine is really important to be activated when we're learning and also when we're stressed. And the absence of it allows sleep spindles to occur between the thalamus and the cortex. Also, the absence of it allows theta to be even larger than it is during wakefulness, and the absence of it allows depotentiation, weakening of synapses. And I'm going to talk a little bit more about what depotentiation is important for. Serotonin also allows depotentiation, so I'll talk about that in a moment. So that theta wave in the hippocampus, which is important for learning and memory, there was a study by Constantine Pavlidis in '89, and another by Huerta and Lissman in 1994 when I was a graduate student at UCLA. And what they showed is if they added to a slice of the hippocampus acetylcholine and then stimulated at the peaks of theta, which are here shown at the troughs of this wave because extracellular neurophysiologists I like to put things upside down. So if you stimulate just 4 spikes at the peaks of theta when the postsynaptic cells are most likely to be able to respond to that stimulus input, you can get long-term potentiation, which is thought to be the building block for learning and memory. It's when synaptic connections between neurons can be strengthened. So beautiful LTP just with one little burst that the hippocampus normally does anyway at the peaks of theta. And then they showed once they'd gotten long-term potentiation, they can weaken those synapses really reliably with just a burst at the theta troughs when the postsynaptic cell is least likely to be able to respond to it. And so you can get a reversal of long-term potentiation, which is cool because it was thought for a long time by computational neurophysiologists that theta trough depotentiation or depotentiation is really important to not saturate your hippocampus. So you need to weaken synapses in the hippocampus because the hippocampus is important throughout your life for learning new things. So here we go. So I knew after that lecture, and I put on the slide that it's a REM sleep-like environment because all they had was acetylcholine, no norepinephrine, no serotonin. So I knew I was going to be going to the University of Arizona where I was going to be able to record from a bunch of cells at the same time in the hippocampus during wakefulness and during sleep. This black box in the middle is each one of those clusters is a different neuron. So you could record from tens of neurons at the same time and have the EEG or local field potential and see where the neurons are firing in relation to theta, both during wakefulness and during sleep. I put on the far right a map of the rat running around on a track, and you can see each of these cells are firing in a different place. Those are called place cells, and it's how the animal learns where it is in the environment and maps in the environment. We didn't have at the time, but I put on the slide also that we put implants later that allow us to optogenetically stimulate the locus coeruleus so it couldn't fall silent in the transition to REM and REM sleep and see what happens to the ability of animals to depotentiate. So the first thing I looked at was, yes, during wakefulness, when the animal is running around on that track, during that theta, which is in red, superimposed on the raw local field potential, you can see that the cells are firing at the peaks of the theta rhythm. Good, because the animals are learning about their environment. So it's great that the cells are firing at the peaks of theta, consistent with long-term potentiation. So at the time I was going there, I knew there was kind of a raging controversy. We'd known for decades from behavioral studies with REM sleep deprivation that REM sleep seemed to be really important for consolidating the things you'd learned, But also there was a kind of hypothetical paper by Francis Crick and Graham Mitchison in 1989 that said, well, maybe REM sleep is for forgetting all those things you learned during the day, like a bunch of screenshots in our mind, things that we don't need to remember, things that are unimportant to retain in our synaptic connections. They didn't have any evidence for it. It was just a hypothesis. But I knew I could look at this stuff at the University of Arizona with my postdoc, because I'd be recording from these cells and during theta and see whether they were firing at theta peaks or theta troughs. And the first data set I looked at during REM sleep, I found that the cells that would fire at the theta peaks during wakefulness fired at theta troughs in REM sleep. So I thought, oh my gosh, this is incredible. Francis Crick is right again. How does he know everything? So there's a burst of activity at theta troughs consistent with depotentiation. Our hippocampus could get saturated. We do need to desaturate it. Maybe REM sleep is for clearing all those memories from, but how could that be? Because we also know that REM sleep is important for consolidating memories. So how could you just be erasing it right away? Well, the next data set I looked at was some rats that came back from space and some other rats that had stayed on Earth. And it was really stable, beautiful recordings. You can record from the same neurons day after day. And what we found is that the cells would always fire at the theta peaks while the animals were running around on the track learning. Good, because they should be learning new things every day. But in fact, it was only the first couple of nights, I'm going to say this is rats, so this is days of sleep. Afterward, let's go back. I just want you to be able to see this forward. Okay. So only the first couple of days after learning the novel maze did cells fire again at theta peaks during REM sleep. And after that, they started firing at theta troughs. This was really cool because Kim and Fanslow had shown a few years before, maybe actually 10 years before, that the time course of consolidation of memories from the hippocampus to the cortex, after which time the hippocampus was no longer necessary, took about a week. So they lesioned the hippocampus bilaterally after an animal had experienced a new environment, one day after that environment versus 1 week after the environment, and tested all these animals a month later. And the animals that had been able to hang on to their hippocampus for a whole week were able to remember a month later. Even if you lesioned the hippocampus, they were able to remember. But if they only let the animals hang on to that memory for 1 day, they acted a month later like they'd never been there before. So somewhere in that period of a week's time, memories could be transferred from the hippocampus to the cortex, after which time it's no longer necessary. Even if it is still used, it's no longer necessary. So the time course of this theta peak to theta trough firing was perfectly consistent with that. Super exciting, and I hadn't anticipated that. But how does the animal know? How does the brain know that a memory's been consolidated? What's that signal that tells the hippocampus it's okay to erase this memory now that it's been stored in its long-term memory structure? One thing that happens uniquely during REM sleep, and this is a paper we are submitting for publication right now, is that REM sleep shows significant backward replay. And what backward replay does is it allows depotentiation. And that's because when the presynaptic cell fires first and the postsynaptic cell fires second, you can get long-term potentiation, but it's been shown for a long time that if the opposite happens, the postsynaptic cell fires first and then the presynaptic cell, you can get depotentiation. And so this backward replay is super important. It really only happens significantly in REM sleep. So this is a really important time for depotentiation. What else do you need for depotentiation? I showed you in a slide a couple times ago. You need silence of the locus coeruleus. So this is a recording that we did in my lab, Kevin Swift, in current biology, that you can see the locus coeruleus during REM sleep toward the right shuts off for long periods of time as compared to wakefulness. So this is the firing rate in active waking, slow wave sleep, intermediate sleep, which is another term in rats for the transition to REM or N2 stage sleep when it's shut off, and REM sleep when it's shut off. It was shown back when I was a graduate student too by Thomas Odell's lab that when norepinephrine is around, when the locus coeruleus provides norepinephrine to the forebrain, it's around, it occupies the beta adrenergic receptor on the cell membrane and causes a cascade of events that prevents depotentiation. That's what that negative symbol to the protein phosphatase 1. Protein phosphatase 1 would cause depotentiation, but it's not allowed whenever norepinephrine is present. So you really need an absence of norepinephrine, a silence of the locus coeruleus in order to get any depotentiation. And that was shown in the right by these two graphs, repeated many times in different studies, that you really can't get depotentiation, which is the white line at the bottom, when norepinephrine is present. You can't get a weakening of synapses. All right, so we wanted to see what happens during REM sleep if you don't allow norepinephrine to not be there. So what we first did is gave animals dizipramine, which is a selective noradrenergic reuptake inhibitor. It leaves norepinephrine at the synapse. Even during REM sleep, it actually inhibits REM sleep. So that's another thing that this antidepressant does. And what we did was we had rats running around on a track and seeing what happens when we switch where their cheese is. This is not actually cheese. This is Ensure in boxes. Ensure, you know, that supplement, they love it. They drink it up. I don't know if you've tasted it. It tastes kind of like ground up Kit Kat bars. It's pretty good. And they love it. So they will run around looking for Ensure in all of these boxes. And there's only Ensure. They all smell like Ensure, but there's only Ensure that they can get when they open the boxes in these three positions. And they learn to ignore the other boxes. But then after they've learned that and they're really confident in running that, we do an evil trick. We switch the place that we give them the Ensure, and they have to now depotentiate the old box positions and potentiate the new ones. When we give them dizipramine after this, they cannot learn where the new Ensure is. Instead, they know that they're not getting Ensure from the old boxes. But they can't remember where the Ensure has been moved to. So what they start doing is just checking all the boxes. So in the middle is a graph showing how much change in that transition to REM N2 stage sleep with sleep spindles, where there's no norepinephrine either. And you can see that the more that we deprive them of the sleep spindle state, the less they're able to learn the new altered position of where the Ensure is. And the less N2 stage sleep they get also, the more sure they are of where, or they keep checking those old boxes. So the less transition to REM or N2 stage sleep they get, the more confident they are in the familiar maze and the less confident they are in the novel maze. So they don't learn to integrate this new information into their map. With the less. So we also did this more recently with optogenetic stimulation of the locus coeruleus. We keep it on during sleep. That's a picture to the far right of the locus coeruleus, a close-up in their brainstem. And it's expressing this optogenetic vector that allows us to just shine light, blue light, on the brain stem and activate the cells whenever we want to. We activate them throughout sleep after learning this reversal maze task, just at 2 hertz. So not strong enough to wake up the animals, just enough to kind of leave it at a quiet wakefulness level, and they get just as much REM sleep and non-REM sleep. And then we do this altered task. And we see that, again, with optogenetic stimulation, which is the line in blue, the more, it's only for 5 hours during sleep, just the first 5 hours that we are activating the locus coeruleus during sleep. The more we do this, and we only do it actually on days 1 and 2, the less they're able to reconsolidate that familiar maze. And the more confused they get. So they're doing about 3 errors per lap toward the end, which is really, really bad. 2 or 3 errors per lap is the equivalent of an animal without a hippocampus or an animal that's blind. The less they also learn that altered task position. We were a little afraid because we only stimulated the locus coeruleus for 5 hours in the days 1 and day 2. You can see that their learning never gets better. As long as we test them, we test 3 weeks on this maze, and they never get better, even though we're not doing anything through the locus coeruleus. So we got a little worried that we had permanently damaged their hippocampus in some way. And so we took them into a new room, and as soon as we did, and taught them a new task without touching their locus coeruleus during sleep, and they learned just fine. So something about messing up their sleep for just 2 days, and not even messing up their sleep. They get normal amounts of sleep, but too much norepinephrine during those sleep stages sort of permanently alters their ability to learn that maze. Not permanently, because actually if we take these animals from their novel task on the far right and put them back on the altered task, they learn just fine again. So there's something about, maybe, I don't know, somebody's got to study this. Maybe they just give up on their map and they just say, forget it, I'm just going to check all the boxes and see how it goes. So so this is what happens during that transition to REM's N2 stage sleep. This is in blue, a sleep spindle in the middle of the graph. And you can see the locus coeruleus is silent for a couple of seconds before every sleep spindle. So that would allow depotentiation to happen. We know that hippocampal neurons reactivate during sleep spindles. That could allow depotentiation and incorporation of this new information and erasure of the old one during that sleep spindle. We know the locus coeruleus fires rapidly in the middle of the spindle, and when it does, it depolarizes the thalamus so much that it can't sustain the spindle anymore, and it stops those sleep spindles. It's cool because when the locus coeruleus does fire, it allows long-term potentiation, so that could be sewing and incorporating the new information into the old schema during the end of the sleep spindle. All right, so this is just an old paper by Gary Aston Jones and Floyd Bloom's labs show that in the second before each sleep spindle, the locus coeruleus is not firing at all. That's 0 down the deepest one there, 0. And then during the spindle, it fires like 2 to 3 hertz long. And we also showed that when we were optogenetically stimulating the locus coeruleus, we got rid of spindles. So the amount of transition to REM sleep, the spindles goes way down when we're stimulating. So it suppressed those N2 spindles. After this reversal learning task, normally with a normally silent locus coeruleus, you get an increase in the power of sleep spindles, so 10 to 15 hertz. You can see the increase during N2 stage sleep. With optogenetic stimulation, it reduces instead of increases. All right. And this is just another way to show the same thing. That sigma band power, 10 to 15 hertz, goes way down. And so what is happening in the hippocampus and in the brain during these various sleep stages? Well, let me just familiarize you. The next 5 slides or so are going to be kind of heavy, but don't worry. Bear with me. After that, we're going to have some fun. So just a few more slides here. I'll show you the circuitry of the hippocampus. The entorhinal cortex, so the entire brain projects down to the hippocampus through the entorhinal cortex layer 2 and goes through the trisynaptic circuit where long-term potentiation can happen really easily in what is now the dentate gyrus. That yellow lightning bolt is long-term potentiation in the dentate gyrus. And then the dentate gyrus gets really excited when something new happens, and there's dopamine and norepinephrine released because the locus coeruleus gets really excited when something novel happens. And long-term potentiation can happen really easily in CA3. And then that projects down into the proximal dendrites of CA1. And then during the N2 stage of sleep, what happens is the hippocampus writes those memories out to the cortex during those sleep spindles. And I'll show you how that happens. And then during REM sleep, that consolidated memory signal comes back into the hippocampus from the cortex that says, hey, we got this now. We know it now. And that memory comes back into the hippocampus through a different pathway called the temporomonic pathway that goes to a different compartment of the same CA1 neurons. And that's the distal dendrites. And it says, and that's where long-term potentiation happens out there, which is how we recognize familiarity. This was Olga Vinogradova, published her last paper in 2001. She did research on the hippocampus in Russia behind the Iron Curtain for her entire research career. And she came up with this idea that the hippocampus has 2 major input pathways. One, which recognizes novelty, that trisynaptic circuit in green there, and another one which recognizes familiarity, that temporomonic circuit in red. She's absolutely brilliant, and I'm so glad that she persisted and did her work and published it so that we could look at it now and build on it. Okay, so Some of these slides are a little weird. Okay, so what's cool about those two different input pathways, the novelty encoding one in green and the red temporomantic familiarity encoding one, is that they are at two opposite phases of theta all the time. So when there's theta in the brain, those two compartments are opposite phases. So you might ask yourself, like I did, how is it possible that neurons can fire at theta troughs when they're farthest away from the action potential threshold and least able to respond? This is why. It's because they are firing in response to that familiarity encoding pathway, which is when the rest of the hippocampus is at theta troughs, it's at its theta peak, so most able to respond. And the two inputs just come in at different times. And when a neuron is able to fire at theta troughs in late consolidation, so it's day 3, 4, 5, after the memory's been consolidated to the cortex, it can cause the cell to fire at what is its local theta peaks, which is the rest of the cell's theta trough. And when it can cause the cell to fire at theta troughs, it can cause heterosynaptic depotentiation. And that is just the same thing as I was talking about earlier when I talked about backwards replay. When the postsynaptic cell is firing and the presynaptic cell is not firing yet, then you can get depotentiation at all those synapses. So you can depotentiate the novelty encoding circuit. Okay, bear with me, I'm almost done, and then we're gonna have some fun. Okay, so why is it important to depotentiate, weaken the novelty encoding circuit once we've consolidated those memories to the cortex? Because you don't want to saturate the novelty encoding synapse circuit with those memories that you just learned once, right? I mean, 6% of our hippocampal cells are involved in learning any new thing. So right now, at least 6% of your hippocampal cells are busy encoding the things that I'm talking about today. And how quickly would we saturate if every time you learn something, another 6% of your cells were actively encoding that? It would be fast. We wouldn't be able to keep our hippocampus learning for the rest of our lives. We would have to erase that novelty encoding circuit at some point. All right, so this is just to show you that during REM sleep, when the familiar memories are coming in through the temporomantic inputs, I flipped the hippocampal neuron upside down here, from the familiar circuits. Without norepinephrine, you can still get long-term potentiation at those distal dendrites, but you can get depotentiation, which is also called LTD, or long-term depression, at the proximal dendrites of the novelty encoding circuit. All right, I'm not gonna just do this. Okay, so this is the importance of sleep spindles. When the hippocampus, which is that CA1 to the left, when it's firing during non-REM stage 2 sleep, the more neurons that are firing in a burst of spikes, the more the prefrontal cortex in the middle will respond and echo that activity in a spindle frequency. So the time between that first burst and the second in red in the middle panel is a spindle frequency spike interval. And so on the far right, you can see the amplitude of the spindle goes up right after the hippocampus is firing. And so that is the incorporation of that new memory from the hippocampus into the cortex, which is listening. It's actually uniquely listening during that spindle state. That is when that new information can be incorporated into the cortex, after which time REM sleep can serve to erase those memories. So a couple of more things about sleep spindles is that it's the distal dendrites, even in the cortex, that are listening to that activity from the hippocampus. There's a huge surge in the amount of calcium that comes in to those distal dendrites, which is consistent with long-term potentiation. And that was studied by Julie Seep et al. And she found a just huge surge during sleep spindles, which is consistent with long-term potentiation. And the only time that happens uniquely in the distal dendrites is during that transition to REM sleep. There's also another cool thing that happens, which is PGO waves, pontogenicular occipital waves. They haven't been studied very much because they're hard to find, because they're from subcirrus neurons just beneath the locus coeruleus. But they, in and of themselves, can cause long-term potentiation, and they also target distal dendrites. So that happens during the transition to REM and 2-stage sleep, and also during REM sleep. So that's when this familiarity circuit can be potentiated. All right, so let's have some fun. Everybody, if you can, get up. If you can't, at least sway in your chair. Stand up. Or I'm going to sing you a song, and I want you to snap along with me because I'm a learning and memory person, so I know that you can remember better through ditties, so I'm going to sing you a little ditty. So I have to go back so you can see the words. If we can hang on to this slide for a few seconds, I'm going to sing it for you. And so I want you to snap, one snap every second. 1,000, 2, 1,000. Good. All right. So this is gonna beat out for us the theta rhythm, so our brains can go 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. Okay, right? Ready? When acetylcholine's high and the repi's low and the serotonin neurons firing real slow and the forebrain's activated by those PGO waves, these create the beat for LTP or LTD, whoa, bidirectional plasticity, whoa, reshaping network, storing memories away to learn efficiently. And now I want you to sing with me. When acetylcholine's high and the repi's low and the serotonin neurons firing real slow and the forebrain's activated by those PGO waves, these create the beat for LTP or LTD, whoa, bidirectional plasticity, whoa, reshaping network, storing memories away to learn efficient. One more time, are you guys ready? Sing it loud. When acetylcholine's high and the repi's low and the serotonin neurons firing real slow and the forebrain's activated by those PGO waves, these create the beat for LTP or LTD, whoa, bidirectional plasticity, whoa, reshaping network, storing memories away to learn efficiently. Yay, give yourselves a hand. Nice job, nice job. Now, even if you don't remember what any of this means, at least you'll remember the tune. All right, well, I have a whole bunch of slides, but you can go listen on YouTube. I give a few of these lectures. Schizophrenia, PTSD, you'll hear a lot about these things at this meeting. So most of these things have been studied in the male brain, so not many of us are looking at female brain, but thankfully all of you who are, because it's very different, the hormonal rates, the locus coeruleus activity is different in females than males, and it might be why we're more susceptible to post-traumatic stress disorder. Our locus coeruleus doesn't shut off during REM sleep in many of the phases, and also why we are more prone to get addicted to opiates and other addictive drugs. So that's being studied by Tanya Lugo. It also probably explains neurodegenerative diseases. The lack of norepinephrine shutoff during sleep is really important to the spread of hyperphosphorylated tau. And it just goes all over. And the activity of the locus coeruleus during slow wave sleep is also super important for hanging on to our memories during that stage of sleep. So yeah, I'm not going to talk about all these things, but just to give you an the last final functions of sleep that I talked about today, which is that the transition to REM N2 stage is for integrating and transferring information, and REM sleep is for cleaning and also lots of potentiation, reimagining, strengthening new. And I hope you guys get a chance to take a nap today. If not, then sleep well tonight. Do not drink alcohol tonight in order to consolidate the things you learned. We know what alcohol does to REM sleep, right? It's not good. Thanks to my lab and my funding sources, and thank you guys for your attention this morning.